Hello students, welcome to EPG Pathshala. I am Mohan Chandra Joshi, Assistant Professor from Jamia Millia and Islamia. Today we are going to discuss about a module named Fluorescence in Situ Hybridization, or called FISH, under the paper Genetic Engineering and Recombinant DNA Technology Part 1. So by the end of this module, the students will be able to understand what are in situ hybridization techniques that are available. We'll get a brief introduction of how fluorescent-based in situ hybridization has been used, what is the methodology that is used to undertake FISH, what are the application of this technique, and in the end we'll summarize how the hybridization technique have evolved and today has reached to the point where it is. Okay, so briefly about how the in situ hybridization technique has evolved over the years, and very initial experiments that were done to visualize how the cell membrane or how different organelles within the cell, as well as how the cellular structure and the subcellular organization of cell is, were done using natural or synthetic dyes. And these dyes, the way these guys used to dye interacts with the cellular structure was that they bind to either protein, nucleic acid, lipid or carbohydrate. Therefore, once you view them under the microscope, you can see the structure at that moment of that particular cell as well as a subcellular organization. However, the major problem with these synthetic and natural dye were that they were non-specific. That means because they bind to everything like protein, nucleic acid, lipid and carbohydrate, it's very difficult to distinguish between which organelle or which particular subcellular organization that dye is staining. Therefore, it was very non-specific way. However, it was a good start. Thereafter, the techniques that were used to make it more specific in terms of in situ hybridization, also to find out within that cellular organization where actually specific protein or the DNA molecule or specific structure is available, is present in a cell. So they used antibodies, and as we know, antibodies are specific to the antigen. So they used antibodies which are conjugated with fluorochrome or different conjugates that were available, so that once that antibody binds to the specific antigen which is present only exclusively to that specific localization, the non-specific were removed, and then by using microscope or other techniques, they were able to localize specifically the organelles or the organization they want to study. Further modifications were made in this technique, and nucleic acid was also directly used to tag with the different fluorochrome and were used to hybridize to the DNA molecule and try to figure out where exactly the DNA is located within the cell. Initially, instead of using the fluorochrome, radio label isotope were used. What it allowed to happen that these radio label isotope was first hybridized to the cell, thereafter was using by using the and they were further localized using the exposure to a X-ray film. However, the major issue with the radio label probe was that they were highly unstable. So they have their half-life, and also the sensitivity is very high because you can detect any emission that is happening. But what happens in that case that you tend to get a poor resolution, because if the signal is very poor, then you have to expose it for longer time. Therefore, you get a bigger signal, and in that you lose the resolution. Also, they were very costly and hazardous because radioisotopes are hazardous to human health. Thereafter, instead of using the radioisotopes, fluorophores were also used for tagging the nucleic acid and were used for hybridization approaches. What it allowed to happen that it overcame all the challenges which posed by the radioisotope. For example, they were safe, they were not as hazardous as radioactive. Also, the turnover period was very high, is very quick, and also they improved the resolution. And prior to using the fluorescent tag, there were other techniques that were also used in meantime. For example, chromomeric reporter. Also, the gold-based probe were also used. In 1980, RNA was directly labeled to on 3' end of the fluorophore to probe a specific DNA sequence as well. Fluorescent in situ hybridization actually allowed to tag a specific region of the chromosome, and also has to track where exactly a specific DNA molecule within the, either in chromosome or within the bacterial nucleoid, is present. And the advancement that FISH has made over the years has made it a very valuable tool for the biomedical research and purposes. Fluorophores now can be added to a DNA molecule with different methods, and also 2 different or 3 different fluorochromes can be tagged to 3 different DNA molecule and can be used for simultaneous hybridization of a cell or a DNA entity, and to find out the spatial localization of DNA molecule of our interest. Fluorescent in situ hybridization is undertaken using different probes, and in terms of DNA FISH, as the name suggests, in this we hybridize the DNA molecules within the cell or within the sample we are looking at. So the very first step that is involved in DNA based FISH is to prepare the probe DNA, which will be used for the hybridization. Now DNA will be first linked with a fluorophore or conjugated with a fluorophore using either Nick translation or PCR based method. In this case, it is very important that sample is processed before the hybridization takes place, because in a live cell or in a sample which are not fixed, DNA probe cannot enter into the cell. Therefore, it is important that those cells need to be fixed by using specific fixative solutions such as paraformaldehyde, glutaraldehyde or formaldehyde, which creates pore in the membrane and then will allow the DNA as a DNA molecule to go and get hybridized with the DNA, with the sample inside the cell. Hybridization process, once the DNA and sample is, when the probe and the sample is hybridized, it can take up to 12 to 24 hours. The next step involved in post hybridization is to remove the non-specific DNA probes which are bound to the cell by using a specific reagent which can have detergent in it and other agents. The hybridization signal is thereafter being visualized by using microscope. Now the microscope can be fluorescent or confocal microscope depending upon our availability and the kind of resolution we are required. There are several methods to trick the system where we can improve the hybridization efficiency, signal specificity, as well as the quality of our signal, and those tricks are usually varies from researcher lab to researcher lab. Here is the schematics of how DNA based FISH is undertaken, or fluorescent in situ hybridization of DNA takes place. So on the left hand side you see the gene of interest which we want to use as a probe. In this case it could be a DNA molecule which is double stranded DNA we want to hybridize. So this method which we are showing is labeling of the probe using Nick translation method, which is a mixture of DNase enzyme, and now DNase enzyme can induce a Nick into the double strand DNA and chew up and create a single strand template, and that template can be synthesized using DNA polymerase. And in this synthesis process, instead of providing DNTPs, we provide DNTPs that are labeled with either biotin or our fluorophore of our interest, fluorophore of our interest. For example, DUTPs which are labeled with specific color of fluorophore. Now, resulting DNA will be a double-stranded DNA in which one of the strand is labeled with this fluorophore. Prior to hybridization, it is very important that that double-stranded probe is denatured and there is a single-stranded DNA has been created. On the other side, meanwhile, when we are preparing the sample, First and foremost, the cell need to fix. That means the cell wall is punctured by using the fixative solution, and the DNA molecule which is inside the nucleus should also be denatured. As you know, a double-stranded DNA cannot hybridize with double-stranded DNA, and when we are using a single-stranded DNA probe, we require a single-stranded template for that to be hybridized. The denaturation of the sample DNA can be done by combination of using chemical as well as the heating method. Once the sample is processed and single-stranded DNA is created within the sample, those are hybridized. As you can see in this picture, single-stranded DNA it will hybridize with a probe which is labeled with a fluorescent tag. Now either we can use a directly conjugate if the labeling is direct labeling, which I'll discuss in subsequent slide. That means that the fluorophore is directly labeled to the probe DNA. We can directly visualize it in a microscope. And if there is a secondary conjugation is required, that means the original DNA which was used the probe, the backbone of that DNA is modified, and now by using a secondary conjugate which goes and specifically bind to that modified DNA will be used for visualization of that specific DNA. RNA fish. Hybridization of fish is, as the name suggests, it is hybridization of RNA molecule within the cell to localize and to identify where exactly the RNA is, and this is one of the latest technology that is being used to find out the spatial localization of transcription during cell cycle, as well as to find out where are the RNA, newly transcribed RNA is localized. In this method as well, we have DNA as a probe, and again the DNA based probe can be made either using PCR or can be made using Nick translational method. This entire process also involves very similar step, but we need to make sure that RNA, the fixative solution as well as the solution which we are using for our in situ hybridization technique in this, which should ensure that they are all RNAs free, and also the salt strength is maintained in such a way so that RNA within the cell is not being degraded. Otherwise, most of the step including fixation, hybridization, washing is done exactly as the DNA fish. So in this example, in this picture, as you can see, the very first process involved is the fixation of the cell, and thereafter the DNA probe is hybridized with the cell, and as the cells are fixed, DNA probes can get inside the cell and it will hybridize with the nucleus or the RNA which is present either in cytosol or inside the nucleus, and then using the microscopy we can visualize and look at the location. So in this picture, as you can see, there is an enzyme, there is a RNA which is being localized within the cell A50, A549 cell lines. As I mentioned earlier, the DNA probes are labeled or conjugated with various fluorophores, and now we have a range of fluorophores that are available with us, which can be used for labeling the DNA probes. For example, in this table we have tabulated all the available fluorophore, has given what are the absorption coefficient as well as the excitation and emission spectra or wavelength for these fluorophore. This is one aspect which we will also dwell little bit in detail in future slide as well as in next module, that fluorophore is a molecule, is a luminescence molecule which has a specific excitation wavelength as well as it has a particular emission wavelength. That means this fluorophore, when excited at a specific wavelength, will go to the excited stage, and once returned to its ground stage, it will emit wavelength of a specific, will emit lights in a specific wavelength. For example, here in this table we see different label compound, whereas coumarins, fluorescence, there are rhodamine dyes, and especially to mention here, there are commercially available very well known dyes, Alexa 488, Alexa 532, Alexa 546, Alexa 568, and Alexa 594. These are the very well known fluorophore in terms of fish technique. Here Alexa 4 represent the name of the molecule, as when where the 488 represents the excitation wavelength. Also we have thiamines, which are another famous dyes, with Situ, Cy3, Cy3.5, Cy5, Cy5.5 and Cy7. Again, they are parallel to or very similar to the Alexa 480 Alexa Fluor dyes, which we have discussed, or which I told you in previous slide, and also have very similar distribution in terms of excitation and emission spectra. There are other fluorophore that are available, commercially available probes, which are spectrum blue, spectrum aqua, spectrum green, spectrum gold, orange, red, and these are again have a very similar excitation and emission spectra. Also we have the stain which are commonly used are for DAPI and the propadium iodide. So in most of the cell biology experiment, where we want to know where exactly the entire DNA is within the cell, so once the cell is fixed, thereafter the entire nucleoid is stained with one dye. In this case it could be DAPI or could be propadium iodide. Both have a different excitation and emission spectra, but these dyes are routinely used in combination with other aforementioned slides. For example, DAPI can be used to look at the to localize where the entire DNA is. Then we can use a specific probe targeting a specific region of DNA, which are labeled with either spectrum green or Alexa 488 or Cy5.5. Now in a resulting image which we see under microscope, we will have a blue background where it represents the nuclear entire overall nuclear distribution, and then a dot or 2 dots, 3 dots, depending upon the localization of the DNA molecules and the number of the site we stain with different color. This is very effective tool, right? Is used by cell biologists to define the cell boundary as well as the DNA boundary. There is another dye which is not mentioned here, is called FITC, which is also lies in the red region, and also been used to define the bacterial cell outline in bacteria. So these are the range of the dyes, different kind of dyes, different kind of fluorophore that are available for hybridization approaches. Now the major part in fish technology or methodology is that how do we label our DNA molecule or the nucleic acid we are interested in with a fluorophore. And there are two methods to it. One is the direct labeling method and second is indirect labeling method. And as the name suggests, direct labeling means when you are directly attaching your fluorophore of interest, whatever the color is, directly to the DNA molecule. So the conjugation that is happening is between the fluorophore and the nucleic acid directly. That's why it's called direct labeling. However, the indirect labeling is, which in that case what happens, first you take your DNA molecule and you attach it with a modified non-fluorescent molecule such as biotin or haptin, and now in the secondary hybridization there will be specific antibodies or through which the fluorophore is attached, and they will recognize these non-fluorescent molecule. For example, the biotin is recognized by the fluorophore label avidin antibodies, and that's why these are called indirect labeling, because the probe is not directly labeled with a fluorophore, but there is secondary conjugate that there is a secondary label which comes in with a fluorophore. This is very important methodology for probe tagging because it improves the specificity. That is one thing which is always important in terms of fluorescent in situ hybridization, that you have higher specificity, and by indirect labeling method we reduce the non-specific interaction, because in direct labeling when you have your DNA molecule directly attached to a fluorophore conjugate, it is likely due to poor hybridization this DNA molecule can non-specifically go and bind to different different region of the chromosome. But in the secondary method, in our indirect labeling method, we are modifying our specific target area through which only a specific antibody can recognize it. That's why it's more specific. And as I mentioned that the probe DNA has to be labeled with fluorophore, and there are two methods for it. One, as I said, direct labeling and indirect labeling. both of these labeling methods are different in terms because they are either directly labeling the fluorophore to the DNA or is indirectly labeling. There are three very well-known techniques for direct labeling: nick translation, we touched upon it in our previous slides, random priming, and DOP PCR. For indirect labeling, there is labeling of probe with aliphatic amines, ULS labeling, one of the most famous commercially available labeling approach, and then we have labeling of protein with protein for direct detection. So we will discuss about these labeling approaches in subsequent slide with more details. Direct labeling method, nick translation. We briefly touched upon this method in earlier slide. We'll go through in detail here. So in this method, the probe DNA, which is a double-stranded DNA, is need to be first stripped off to form a single-stranded DNA. So in this case, what happens that we use a combination of DNase enzyme. DNase is the enzyme which introduces a single-strand nick, and also a DNA polymerase enzyme which synthesizes the DNA. So when a specific ratio of these two enzymes, which is optimized depending upon the length of the probe DNA, and is optimized also in terms of the time incubation time that we are keeping, we are treating our DNA with DNase for long. If we keep it for a long time, the entire DNA can be chewed up. So DNase 1 creates a 3' free hydroxyl group. Now once we have add DNA polymerase to it, it adds that it will add a nucleotide to the 3' hydroxyl group. Now because we are providing the labeled fluorophores in this case, for example DUTP, which are labeled with different fluorophores, while therefore when DNA polymerase is synthesizing a chewed up single-stranded DNA, we will incorporate these fluorescently tagged DUTPs. And remember, we cannot incorporate all DNTPs which are labeled. That means that all the DNTPs which are used by DNA polymerase to incorporate post-DNase treatment are not labeled with fluorophore. There is only one, either DUTP, which is labeled with a fluorophore, is tagged. Therefore, the distribution of fluorophore tagged with the DNTP tagged with the fluorophore is not overwhelming. It's 1 or 2 base pair in every 100 nucleotide. Random priming, the other direct method of DNA polymerization of DNA probe formation, and as the name suggests, it requires random primers, which are actually nothing but a combination of hexamers, octamers, and nanomers. So what happens in this case, we have a DNA template which we want to create a probe from. This DNA, double-stranded DNA, is denatured and then mixed with oligos, which are hexamer, octamer, nanomers, and because these are randomers, so they go and they act as a primer and they bind to the non-specific areas, non-specifically to the template. Once we provide clino DNTPs and are labeled fluorophore, so the DNA reaction, the DNA is synthesized because these oligos works as a primer. Subsequently, what happens, similar to the nick translation method, we'll have a double-stranded DNA in the end, in which one of the strand is labeled with conjugated fluorophore DNTPs. This can be denatured and used for subsequent hybridization process. The third method is degenerative oligonucleotide prime PCR. In this case, instead of using the random oligomers, hexamers, octamers, we use primers which are degenerative. For example, instead of having a D or T at a specific location, for example, we can have R, which is shown in this table. So that means that R can be either A or can be G. Similarly, we can have H instead of a DNTP, which can represent A, C, or T. So in this case, so by doing this, the degenerate primer will have a combination of nucleotides, which could be either A, C, G, T. Thereby, the amplification is non-specific. But in this kind of PCR, the first step is low fidelity. That means so that the degenerate primers are allowed to bind to the template. But after a while, the second round of reactions or the second cycle is high fidelity. That means they are kept at the higher temperature, so that once the template is amplified using the low fidelity using these degenerate primers, subsequently can be amplified by subsequently amplified at a higher temperature. Now, indirect labeling method, as I mentioned earlier, indirect labeling, in this we modify the DNA template which we're going to use a probe, and then use a secondary labeling method to detect it. So it's a two-step process. In very first step, we use DNA polymerase enzyme, which incorporates a nucleoside triphosphate into the DNA that carries a aliphatic amine modification. Now in the step 2, we incubate it with a modified DNTP, through which this amine active fluorophore reacts, and resulting in a DNA molecule containing an aliphatic modified base. This method allows labeling of range of DNA molecules. Range of molecules, it could be DNA, it could be RNA, it could be proteins as well, and has been used for various applications. ULS labeling. As I mentioned earlier, these are commercially available indirect labeling methods. ULS is very well known, and enzyme which is universal linkage system, and it is a platinum based dye system that interacts with guanosine of the DNA at base number 7. So the interaction results in a stable bond between nucleic acid and platinum fluorophore complex. For this, ULS based labeling method can be used for various DNA molecules, including plasmid, cosmids, and high molecular weight DNA molecules, such as DNA in low melting point, agarose, bark, yak, whole chromosome library, other DOPPACR product. So it has a very large range of DNA substrate. However, the important and the crucial factor in this method is that we need to optimize the DNase treatment or the DNA fragmentation method, which could be achieved either by DNase or by sonication, because we need an optimal size or optimal length for the DNA molecules that can be used for the subsequent reaction. So one of the major application of fish based method is in karyotyping, and this is what has revolutionized the cell biology and the human genetics research. And as you can see in this, the entire chromosome, whether it is chromosome number 1, 2 or 3, can be karyotyped, means they can be painted with one single color and can be visualized in such a way. So this technique is very important in terms of understanding the genetic disorder or deletion or translation or insertion that has taken place across chromosome. You can also see if any fragment of chromosome number 15 has translocated to chromosome number 10 or 16 for that matter. So this method allows us to understand the genetic aberrations, any chromosomal abnormalities that are generated, and this technique is often used for screening the genetic deformities in the neonates or during pregnancy. Another application for fish based method which has also evolved is to determine the pathogenic infection in bacteria in humans or not. So we can figure out whether a person is infected with a specific bacteria or a specific bacterial pathogens or not. Though this technique is now not been oftenly used, because there are sequencing based methods which are easy, faster and cheaper, but still fish based identification of bacterial pathogens is one of the application which has still been used in this case. So we can collect the specimen from the site of infection or from the place where we want to investigate, Then we synthesize the pathogenic specific probes. We can hybridize it just like I've mentioned in earlier, and then we can visualize it in a microscope by using fluorescent microscope, and figure out whether that particular pathogen is there in that specimen or not. One of the major application and through which this cell biologists are pushing toward is to use of this technology in understanding how chromosome is organized, whether it is in eukaryotes or also in bacteria. As you can see in this picture, on the left hand side you see a metaphage DNA is painted with different colors, and this is achieved by using in situ hybridization of FISH technology, in which the entire chromosome is painted with a single color. Now what this would allow us, the researcher will to do, is to understand how these chromosomes are organized within the cell, also to define the area where there are two chromosomes are interacting and where are the chromosome territory. This technology allows us to figure out where are the boundaries between chromosome X versus chromosome Y, or chromosome number 1 to chromosome number 5, and also the result has also allowed us to figure out whether which region of the chromosome can be transcriptionally more active because they are present on the chromosome territory. So the application of FISH technology in understanding the chromosome structure and organization is immense. And also on the right hand side you see another picture where it shows in terms of a single dot where we have labeled a specific region of a chromosome by using FISH technology, and you can see where within the cell where exactly that signal is getting co-localized. This technique is also now been extensively used in bacteria to understand how chromosome in bacteria is organized. As you know, bacteria is having a single chromosome, so the different region of the chromosome can be painted very similar to what we have seen in this picture. Also been different part of the chromosome in bacteria is painted with different colors to see how chromosome is organized. And by doing a time-lapse experiment in which we captured the cell at different cell ages and painting them with this technique, we can also see how chromosome domains are being reorganized during the cell cycle. So this technique is very powerful and been extensively used by cell biologists to understand the chromosome structure and organization. So in the end, we summarize the entire module. So in situ hybridization techniques have improved the resolution as well as target specificity. Apart from fluorophore, gold particles, radioisotopes has been used earlier for hybridization technique. The range and type of fluorophore that are available for us has also increased the applications of this hybridization technique. Now we can use multiple fluorophores to locate within a small region of DNA or protein as well as. And this technology has been a game changer in biomedical research, as I mentioned in earlier. Also, by karyotyping, we can also figure out what are the possible birth defect in a newborn, in a fetus, as well as also we can use this one to figure out how many deletion insertions are there in the chromosome. And also using this technique has been FISH has been, as I mentioned earlier, also in both bacteria as well as the eukaryote has been used to understand the chromosome dynamics during cell cycle. So this technique has evolved and has given a lot of opportunities for researchers to dwell in different area of cell biology. Thank you.